One.
Welcome to The Cutting Edge with Dr. Lee Pierson and Dr. Robert Stubblefield. I'm Steve Richins, and today on our show we have a special guest, Dr. Robin Mockett. And our topic is anti-aging research coming of age? And I'll hand it over to Lee.
Okay, so we're going to it's going to be mostly Robin today, although I'm going to be posing some questions to him later. But I should say Dr. Mockett, because Dr. Robin Mockett is a gerontologist. Nowadays, we usually put the prefix biogerontologist to distinguish those people from the sociologists who study, the effects of aging on your family and stuff like that. Dr. Mockett is a real, I call him Robin because I've known him for many, many years. Dr. Mockett is a real, he-man biologist. Oops, he could be a she-woman biologist too, but he happens not to be. So he's a real biologist. He does bench work, lab work, and he'll tell us about that later. And he is an associate professor of biomedical sciences, I believe. That's nice. That's a nice title. Covers a lot of ground. And Dr. Mockett is going to start off here and give us something of a brief overview of the situation of the field of anti-aging research. So Dr. Mockett, take it away.
Okay.
Oh, and it's starting with a disclaimer, right? You want to start with a brief disclaimer. Go ahead.
Yes. Thank you very much. I don't quite know who I'm talking to, but I understand that I am talking to a recording that may be listened to by anyone anywhere in the world for the entirety of the future. I haven't done that before, and so I think I need to be a little bit careful for a couple of reasons. One is that in the course of doing a lot of teaching these days and a lot of bench work, that I spend less time in the library than I would like. And this is not as rehearsed as I might ideally expect it to be. So it's a little bit off the cuff. The other reason for hesitation is that I am supposed to talk to some extent about my own research, and within the research field, there's a custom when we go to conferences of not sort of broadcasting everything to the whole world because of the issue of pre-publication, which is to say announcing your results before they're reported in a scientific journal can interfere with that publishing process. And so I expect to be more fine grain about my own work when we go into the after discussion that isn't just recorded for everybody to listen to indefinitely. So if there are people out there who want to hear some of the particulars in a finer grain, what's really going on in our lab today and this week and this summer, some of the fine grain I'd say for afterwards.
From the clubhouse.
Yes,
that's clubhouse. Yeah.
Yes.
Okay, very good. Very good.
And I am always receptive to conversation as time allows. So I think my university address is out there for all the world to find on the South Alabama site. But I can also go into private email conversations with people. I maybe shouldn't put my personal email just like for literally the entire world, but I'm very receptive to sharing that through the chat on the clubhouse and so forth. So, so I want to give overall my perspective. If I'm being autobiographical, I'm 50 years old. I got interested in trying to live a long time and change the rules of life and death around about the age of 4, once I understood what death was. And I think there are a decent number of people out there, both in the field and as subscribers to a lot of the newsletters around the field, who can relate to that. And so a question that will come up for many people, depending on audience, is what can I do to make myself live longer? And so in general, Dr. Pierson Lee will probably attest that he generally encourages me not to depress everyone too much, because on balance, I think this is a field that has been just right around the corner from the elixir of life for at least 3,000 years. And there are grounds to be encouraged by the things that are being done, but there are always grounds to be encouraged. And we don't yet have a track record of people living longer because of anti-aging research showing up in terms of the oldest people in the world being people who have got to that age because they took interventions. So if we're looking backward, or as best we can tell at the present, we aren't currently slowing down our aging process right now as a result of the work that we have done. And we can be, and to an extent, I encourage optimism, but I myself am not taking any anti-aging interventions, and there aren't any that I recommend at this time. I hope to recommend some in the future. I've always come at this field with the belief that we can achieve a success. I would define a success as meeting somebody, shaking hands, and needing to ask, are you 20 or 120 years old? And not being able to tell by looking at the person. That would be a good benchmark of a full success in anti-aging. I think that's achievable, just the same as splitting an atom and having the horseless carriage, the car that drives without an animal pulling you. In terms of whether it would happen in a lifetime, I've often been asked to make predictions about that. I find that they are speculative enough as to be of limited value, but they may serve either for entertainment or just like as a guess, but they shouldn't be taken as an authoritative guess. If I give those guesses, when I started off thinking about this, and at 7, 8 years old, 1980, I figured you probably need 3 80-year lifespans back to back to get to a success. So I would have said we'd come to a success point in the 23rd century. When I got into the field in the 1990s and started doing the genetic research, making transgenic flies, studying the oxidation theory, that was my graduate and postdoctoral work. I thought things were going amazingly well, and I thought it's conceivable that this could actually work out in time to make my life longer if I live to an old age, which would say 2060, 2080, somewhere around there, we'd come to this stage of just being able to control and stop our decline. In the 2000s, I found the progress of the field was quite dispiriting. There's been a move toward the small molecule magic bullet type treatments, which I think give a lot of people hope and haven't been so encouraging to me. In the latest decade, I've become more hopeful again, and a large amount of that revolves around the advancement of our capacity, not directly the work being done in anti-aging, but the capacity to do tremendous amount of sequencing to get information that wouldn't have been available before, related to things like the variation of the DNA content among the individual cells within our own bodies. That sort of thing has really a capacity to understand what's going on in the body and to get at what is the problem and solve it. But I come with an orientation of expecting to need to understand a complex, elaborate problem and try to design a rationally targeted solution to that, as opposed to getting lucky with a simple chemical fix. Especially chemicals that already exist in the body or that we can get through the diet, given how long people have been studying and how much variation there is in the things people eat and that they do. I think a transformative discovery that was going to come through those methods would already have been achieved by now. So I think it's a more complex engineering type problem rather than a simple fix. And the pair of companion papers by Alexander Wolff, who I do not know, but who I appreciate a great deal after reading his papers in Mechanisms of Aging and Development from last year, that helps to give a good overall perspective. The last general thing I want to say, Lee has given me a list of 10 topics to comment about. I have
I was going to go over those basically one by one, talk about those one by one.
Yeah, I have That's great. I have superficial thoughts on almost all of them. I read a little bit on that last one, which was your first one, the Yamanaka factors. I just glanced into that a little bit last night. But the last general thing, may I do a last general thing for the people, please. The people who want to say, what can I do now if I'm not going to try and sell you a pill and I'm not going to recommend any of the pills anyone else is selling? I do want to put a few really basic, staid, boring, grandfatherly kind of thoughts out there that people may overlook, and that relates to this excitement at the idea of living to be 140 instead of 70 or 80. Make sure to get the 70 or 80 years that are comparatively easily obtained without doing anything special. So I have 3 things for that, especially for the youngest people. If you look at what kills somebody young, and I'd say anything less than 70 as an unlucky premature death, you've either been unlucky or you would hope hope you could have prevented if you die less than 70. So my first thing is, if you're 20, 18, 25, the number one danger of you not getting the 70 years and coming to a point where we have tremendously more knowledge, so that anything we do to intervene either will more likely work or be safe, then the number one thing is very simply be careful. The suicides, murders, accidents, especially vehicle crashes, the alcohol, the illicit drugs, all those things are the big danger. So just simply don't be reckless and lose all of it at the outset. That's the thing for the youngest people. For those moving a little bit on, I guess we'd be talking, really all of them are for everyone, but more for 30s and so forth. You start into saying, well, my great aunt lived to be 104, so I've got good genes. Ask how old did your shortest lived 2 or 3 direct relatives live, and ask what was it that caused them to die young. Is that something that is truly unlucky, or is that something that I should keep an eye on and monitor whether I'm at risk for those things myself? Because to stay alive, everything in the body has to be above a certain base threshold of viability. And so this metaphor that the chain is as strong as its weakest link really is applicable in staying alive. When it comes to genetics, the people who've studied how much does your length of life depend on how long your relatives lived, when they survey the entire population and how long people live and relate it to the life lengths of relatives, they find overall about 15% of the variation in one person's life is reflective of how long their relatives lived. And that's largely clustered in a few people who have unlucky genetics that lead to a short life, and a very few who are in families where there are a lot of siblings who cluster together living into their 90s or centenary years. So your best predictor of long life is your siblings, because they're the ones who share all of your ancestry. But that would be my thought related to genetics. Look to your shortest lived ancestor and expect some association of longevity with genetics, but a little bit minor issue for most people. And the third one of 3 is related to this notion of the chain and its links. And that will apply especially, I'd say probably for everybody or nearly everybody at 50 and onward, is you probably already have something that you know is a little bit something that you're not the Iron Man or Invincible Woman in some aspect of your personal biology. And so my advice is guard your weaknesses. Whatever is the thing that is a vulnerability for you personally, be careful not to challenge that thing with extra assaults or damage.
Okay, sounds good to me. Watch out for those weaknesses. All right, so I've got, we have a list of things. I did want to mention just a little bit about that Wolf article, at least the first one, that not every, some people might have read it because I posted it on a reading on HBL actually. But just to say a word, this fellow who I've never heard of either is in Germany, and his basic intention in the first article, at least, as I read it, and Rob and Dr. Mockett can correct me if I'm wrong here, is that caloric restriction and the various caloric restriction mimetics, things that do the supposedly chemical interventions that do supposedly similar things, that rather than actually being lifespan increasers, they don't actually increase lifespan as such, or they do increase lifespan in obese animals. And Wolf's contention is that generally speaking, the animals, the mice or rats, rodents used in this area of research are obese. They feed them too much in the lab. So both the control groups and experimental groups would be obese, except that these chemicals reduce the weight in the experimental group. So that basically the effect is to show that all these substances have some kind of weight reduction function rather than life extension per se. And that's a pretty serious, if that's true, that's a pretty serious knock on the whole field, a lot of research in this area. And let me take some of these things one by one, and I'll ask Dr. Mockett for just a quick reaction. You don't need to do any deep analysis, which would not help us anyway. But the field recently has been invaded by zombies and vampires. So let me bring up those things first. This is actually my favorite of the near-term interventions: senolytics, substances that kill senescent cells. And senescent cells are cells that, as you age, I'm not just going to give you the briefest description. They lose functionality, but they stay around and use up resources and also pump out various toxic substances into surrounding cells. So even though it turns out they have a function, they're also implicated in aging. And there are substances that kill them, such as quercetin and dasatinib, a cancer drug that was a combination, and ficitin, another polyphenol related to quercetin, is even better than quercetin. And this to me seems pretty good. What do you think, Robin? Is there any hope in this area?
That's one of the ones that I think is encouraging. I think it's definitely an area where it's worth doing the research, and it's worth reading the research when it comes out. So I feel encouraged about that. They aren't one of the ones that's mentioned in Wolf's first article, right. And they almost force us to say something about the second article. Which is what he thinks is really going on in terms of aging. And I think a quick summary of that would be that our fundamental problem is that DNA is just an inescapably unstable molecule that is susceptible to damage and mutation. And when the damage occurs, there's a capacity for repair. Usually the repair will be correct, usually meaning 99.999, maybe more 9%, but there are just vast numbers of damage events that occur, as in tens of thousands every day, and that some of the damage gets repaired incorrectly. And when a cell divides, you have to get rid of the damaged adducts in order to complete the cell cycle. And if you do that and you get one of these errors, then you have a change of the sequence that is actually otherwise silent. So you now have a fully regular DNA sequence, but it just differs from what was there before. And so that effectively locks in mutations. And so the problem then for us in aging is that the more cell division we have, the more these mutations occur, some of which will lead into loss of regulation of the cell cycle and a gain of function resulting in cancer. So in general, the fear with mutation is not that it's going to make the cell inactive, it's going to make it that it divides divides too much, overwhelms the population of cells, and just runs amok, and we get cancer and we die of cancer. And
all right, let's go on from there. I think that's a good summary of what Wolf said. Or we want to add one more thing to it?
Yes,
we got a lot of little points here to cover. So
yes,
go ahead.
That's half of his point. So then the second half is that mechanisms are put in place to stop that by evolution and in our nature, to avoid everybody just dying of cancer, or to delay that as much as possible within the framework of what repair we can do. We have the ability to stop these cells from going out of control. So either you can get them to just die directly by apoptosis, or you can get, in the case of stem cells, which are replenishing, they're the pools for replenishing all our cells as they get damaged and need to be replaced. That if a stem cell undergoes this kind of damage where it could cause a cancer, then it gets rooted into differentiating and shortening down its telomeres and running down its capacity to keep on and on dividing. And
okay,
so
I think we better. I think we're getting to the point of satiation there on that. But basically, what happens is a lot of those things cause cancer, and we have
the point is that these
proliferation.
They prevent cancer by causing things like cell senescence, which then cause aging instead of cancer. And with the senescence, then that is the basis for the word senolytic, is that these cells that have become cellularly senescent and then are not capable of flaming out into a cancer.
And we can kill them. We can kill them, even though evolution there's no natural selection pressure to get rid of those things in old age. We, as conscious, rational beings, can figure out how to kill them, and that's being done now, and that's a good thing and may extend lifespan.
Yes, I think that is fair. That I would not say that is going to work, but I'd say that's a very reasonable hypothesis, a good framework, something absolutely that should be tested, and people are testing it and getting encouraging results.
You mentioned stem cells. How about stem cells as for example, the new research in iPSCs, in these cells that are where you take old cells and return them to having different capabilities.
Right,
and then
I think
use those to repair. You can repair organs with those kinds of cells once we have control of that. What do you think about stem cell therapy?
I think that is hopefully a passport for us. I think this is the sort of thing that can work. The cautions I have with these two things, with the senolytics, the concern is that if you have too many of your cells being senescent and you go in and kill them all, are you left with just no cells left? So I mean, if I look at myself,
my understanding is that the number of senescent cells, even in ancient people, is fairly small. The percentage, is fairly small. That's what I hear anyway.
Yeah, the truly inflammatory senescent cells, that's right. And so, as with the tumors, the concern is the various stages of how far are they on their way to senescence, this kind of thing. This is a caveat. This is where it's necessary to just go and really be cautious and not say, we're going to take one step and problem fixed.
How about stem cells?
The stem cells, I think it's a very, very promising thing. I would like to be 20 rather than 50, and I'm thankful I'm not 80, because I would suspect that the first forays in that field are going to result in our learning more about the cell cycle that we don't fully understand. Because when we try to introduce that, when you introduce cells that have a great capacity for division, the capacity to introduce something that produces cancers, that's a real one. So I expect the field to have to go through growing pains where the first treatments turn out to be more dangerous than we thought at the time.
That's already come to pass. That has already come to pass in that field some years ago. All right, let's move on, because time is moving on. And I want to eventually get to your stuff too. So I'm going to kind of accelerate things a little bit here. Gene therapy and gene engineering, that general area. What about that? Changing the genes, changing the expression of the genes, sending genes and altered genes into cells with viral vectors. That's all that sort of thing. There are various ways of doing that. How about that sort of gene therapy, gene engineering area? What do you think?
Very similar to the stem cells, and in fact quite overlapped with it, because one of the plausible ways to do the engineering is to take out some cells, engineer them outside the body, and reintroduce them, because that's going to cut down on the challenge of saying, well, how do I get my viral vector past the blood-brain barrier and down into a tissue and engineer the cells that are deep within? Of course, if you take the cells out and then you try and reintroduce those, you also have to get them into the right location, which I think will be easier for some locations than others. And safety, again, is going to come as an issue. So the CRISPR-Cas system is being used for making these very specific gene edits. And it seems to be very safe and specific, but the concept of off-target effects arises where you get that there's a different genetic modification somewhere else in the cell. And so I'm seeing, now don't have time to read all of them, because reading one paper is about 4 hours in some of these fields, and there are 1,000 papers every day. So, but with the CRISPR-Cas system, there's a good amount of work being done very actively to produce alternatives and variants that are safer in terms of eliminating these off-target effects.
Okay, I'm gonna move along here because time is moving on, Dr. Mockett.
Okay.
Telomeres. Telomeres.
In the
90s, the little caps on the end of, just for those who, I'm sure everybody's heard of this, but the little caps on the end of DNA, and every time your cells divide, a little piece of this is snipped off, and therefore eventually they start getting frayed like the ends of shoelaces. And so there are substances that add bits and pieces back onto the telomeres. What about that? Is that of any use to us or not, Dr. Mockett?
It's intertwined. In the 90s, when I learned about the telomere theory, I thought this was going to be totally a misdirected line of effort. And my view of that has changed maybe 5-6 years ago. The reason is that the problem is that when you divide a cell, you copy the DNA. You can't copy the very last bit at the end or the very first bit at the beginning of the DNA. So you lose a bit every time the cell divides. And if you do that too many times, a few dozen, you come to the Hayflick limit, which is that cells won't divide anymore because these protective endings have just been eroded away. So there's this enzyme telomerase that can add pieces back to replenish those ends. And so I was initially of the view that that wasn't going to be useful in aging, but would be useful in cancer, because a lot of the manifestations of aging were in things like heart muscle and neurons, which themselves are not dividing. They're terminally differentiated. They shouldn't have a replication problem. The reason I switched back to being interested is because of the notion that there's so much need to regenerate our tissues through regular duplication of the stem cells. And so then the issue of needing telomerase to be on in order to avoid those cells coming to a limit, that is an important potential problem. So for that reason, I think telomeres are something that we do need to have in mind. It also, again, relates to the balance that if you turn the telomerase on, the cell can become cancerous. If you shut that off to stop cancer, you run the risk of aging. It's this opposing jaws of cancer and aging over and over again.
Yep, very good. We talked about senolytics a little bit, that those are the killing off of zombie cells. Then there's another proposed intervention that's been studied in rats, maybe other animals, that is a form of, you might say, vampirism, parabiosis, where you take two rats and put them together and meld their circulatory systems so that they're circulating the same blood between them. And the young rats, briefly, the young rats get old and the old rats get young, to put it crudely. What do you think about parabiosis?
That one, the groundwork keeps shifting, including it feels like maybe a week or two, but maybe it's already a month. The idea was that, okay, if you get young animal blood and you circulate it through the old animal, that it appeared that the manifestations of aging in the old animals disappeared or diminished. So it was good for the old animal. And the question also then came, is it bad for the young animal because its blood is getting dirtied up? And so there was a discussion or debate or investigation, really. There were investigations to find which of those things were the case. Initially, the reports I understood were saying that the young blood was pretty good for the old animal in being possibly a supply of chemical factors that then we could hope to identify and deliver without harming a young animal in the process. Steadily, my understanding is that the ground has shifted in the direction of saying that the old blood actually has more harmful stuff in it that has come probably in fair part from the senescent cells. And I've also in passing come upon reading that some senescent cells can actually induce other cells to become senescent. They can have a domino effect. But it seems now the latest one I came across, but really just in passing, was that the old blood was harmful to the young animals more than there was a benefit to the old from the young. And so that more you would hope to be purifying harmful stuff out of your blood as an older person or other animal, which could be worthwhile.
Which is hopeful.
Too.
That's hopeful.
Yes.
And it also reinforces the idea that working on senolytics is a good thing. reinforces that as a possibility.
It
Okay, I'm going to
becomes
move.
like, effectively, it
Go
would
ahead,
be
one more thing.
analogous to dialysis.
Yeah, okay. All right, how about, I guess this, well, I'm going to say the mTOR inhibitors. I guess we haven't got a lot of time, but I want to talk, we can talk about that when talking about your research, because you studied rapamycin, right? But mTOR
Yes.
inhibitors, how about, so I'll save that, that's an important one. How about NAD plus and its precursors like nicotinamide riboside that some people are touting heavily these days?
That's one of the ones that's listed in Wolf's first article as something
Right.
that is promoting weight loss by facilitating greater energy utilization, which could be good for those who are overweight. I do want to interject here something in the vein of just like being helpful for people like in the current on a low tech. I heard this in a few conferences, and I think it's a valid thing to share. And that is the idea that through most of our lives, we're taught you don't want to get overweight. It's harmful. It makes you have a shorter life, which I absolutely think is right in a context where the overall population weight has got bigger and bigger and bigger over several decades. But when you get to older adult life, probably even the BMI guidelines, there's discussion they will eventually likely change in favor of saying that you would like to be a bit bulkier after about age 65, that your main vulnerability to death comes from frailty and loss and lack of strength. And so if you have some bulk that will serve as an energy reserve in the case of a viral infection or some digestive problem, that is actually healthy. And also in terms of things like mobility, that the muscle strength needed to carry a bigger body, it's actually better to have more self at retirement stage. So I've taken to discouraging older friends and relatives from dieting when they're in the senior years, trying to get down to the young, thin size.
I certainly would like to hear this, but I'm not going to buy it 100%. But I was certainly I am enjoying hearing this.
Yes.
Robin.
So what it means is that if you're at a reasonable size, or if you're at a constant size, if you're 60, 70, 80, and you haven't just had a recent great big gain of weight, you're probably best off to be preserving the weight rather than trying to lose it. And so any of these things that are for making your energy reserve get burned off and used up, they could quite plausibly be undesirable.
Okay, I'm going to move on here. This time marches on. How about lipofuscin removal of the garbage products of cells that are thrown into little what do they call them? Anyway, little
autophagosomes, perhaps.
Perhaps,
so,
yes.
Yes.
And that those get filled up, and this is not good. What about lipofuscin removal? What do you think?
Right, confused with the
do, and can we do it?
I've done, and I'll say this as a component of my own research over the past 10, 12 years. I hate the piece of equipment, by the way. I think it's hemorrhaged several years of my life, this rotten piece of equipment that's just almost good enough, but not quite. But for my studies of the fruit fly lipofuscin, it looks to me that there is some increase of lipofuscin during aging in those animals. It's difficult to measure by the method that I've tried to employ. It looks like there's a fair amount of variation among the animals, and so with the universality of the aging versus the lipofuscin buildup being quite heterogeneous, that's a little bit concerning for the theory. But broadly, the theory has been that aging is the result of damage to molecules that doesn't get repaired properly, and that you can facilitate the repair. The basic mechanism is that you self-eat or autophagocytose the damaged stuff. You fuse it with lysosomes, you degrade it, and now you don't have damaged stuff. But not all the damaged stuff can be removed.
But can we do this? Can we do this now? Can we do this? Not ourselves, which can do it to a certain, it's a certain degree, but the places that the stuff goes get filled up. Can we do anything ourselves about this issue?
So there are two possible questions here, and one of them is, can we remove the stuff that is the lipofuscin proper, the true lipofuscin that is left behind in lysosomes after autophagy? I'm not aware of a way of removing that right now.
Okay.
And the other one is that you have damaged stuff distributed, and you can clear away the non-functional stuff by putting it through autophagy, running it through the lysosome, and removing most of the damage, and then just keeping the worst.
Can we do this? Can we do this now or soon?
Can we facilitate our autophagy?
Yes, yes.
I believe that we can, and this is the whole idea around rapamycin, or is certainly a major component of the idea around rapamycin and mTOR, is that if you have the TOR complex that is active for cell metabolism in times of abundant nutrients, that if you apply rapamycin or some other mechanism to shut that regulator off, that then the cell will perform autophagy and degrade some of its own stuff, and in the process it will clear out damaged stuff that might have built up. So, okay,
I'm going to move on here because I got a couple more quick ones that are important. Epigenetic reprogramming, that is changing the way genes are expressed by changing the way the shell, that the histone, the part of the stuff that is the superstructure of the DNA, and changing that so that the way DNA are expressed is changed. And there's been recent work on that with what are called the Yamanaka factors, and David Sinclair, who some people like, has shown that you can use, by using 3 out of 4 of the factors, you can avoid cancer in some interventions. Is this a good area?
Okay, so out of the 10 things that we listed off a couple of days ago, this was the one that I had least knowledge about and was least prepared to talk about. I've looked just very quickly last night to refresh myself about that. I see that I had read something about that a few years ago. The notion of epigenetics and having the epigenetic marks will give you a biological clock of how old you are right now as an indicator. It seems those clocks sound pretty good for that purpose. I can reasonably believe that you could adjust those things. I don't know yet that I feel confident that those are causing the aging as opposed to being just a marker of it. I'm open to that being a useful line of research, and again, something where this cancer versus aging balance is going to come into play. As for the histones, insofar as they would lead us into the sirtuin and resveratrol topic, that's one which was one of my causes of discouragement through the 2000s, because I didn't attach
I
much
left that
value
off.
to
I
that.
left resveratrol off because that's been pretty well scotched in the last 10, 15 years. So I left that one out. But I got one more. I don't know if I should broach this one or not, but I'll what about the SENS approach of Aubrey de Grey? The idea of there are these 7 areas and you just go and fix the damage. And then if it comes about again, you go fix it again. So you just do that. What do you think about that? In general terms, I mean, obviously that's a whole big subject in itself, but
I think it's a good framework for listing off and identifying the kinds of destruction that occur in the phenomenology of aging. And I think it really helps us to see the complexity of the problem that we're facing. So the idea that many kinds of different damage to all the structure of our cells is going to be easily fixable by tweaking one gene or taking a different chemical. I think that just reading what all the SENS damage is should show how much it seems just tremendously optimistic to think that there'd be an easy fix to aging. So I recommend reading that just for that reason. And in terms of whether the fixes have to be piecemeal, in concept, certainly we could step back and get something that doesn't need 8,000 fixes for 8,000 problems, because you can do some preventions, but I think it's a good framework for understanding the phenomena of aging.
Okay, let me interrupt you. We got a couple of super chats. And
all right, let's
thank
go for
the
that.
Ayn Rand Center UK for sponsoring this, and and share and all of that. And thanks, Dr. Mockett, for coming on. So the quick questions I have are from, the first one is Friend Harper is the name.
Yeah.
And for $4.99, they say, great content and important stuff. The science is coming along. I just hope the culture doesn't try to get in the way of it. Sorry, that's a comment. This second is a question.
And he's right. That's already happening. This is not an answer, but a comment on the comment. I can comment with 3 letters, FDA. No, I won't say any more. Go ahead. Next, Steve.
Okay, the next question is from Apollo Zeus for £5. Have you thought, and Lee already mentioned David Sinclair. So his question is thought on the work of David Sinclair. And then he thanks you, Robin.
Okay,
So,
I turned that over to Dr. Mockett. Hot potato there, Dr. Mockett. Hot
yes. Dr. Pierson has, in a quasi private discussion group, but still with several unknown people, shared some of my off-the-cuff one-on-one comments, so I wouldn't want to share them for the whole world. I will say that I don't know Sinclair. I know his style just through the interviews that I've read and the articles, and in terms of personality, we seem very opposite. In terms of the anticipated pace of discovery and the level of confidence we can and should have either in taking an intervention now or in saying what's the percent chance that so and so is going to work. I'm usually going to come down as a cautious maybe or probably not in general. And so stylistically, I think that we're very, very different, even though I don't know him in person. The sirtuin and resveratrol stuff, I think historically was one that he was a strong proponent for, and I didn't think it was very promising, and it doesn't seem to have been. I'm not as up on what he's doing now, but to the extent that it relates to NAD, and there are people who've been involved with NAD and energy, just the entire genre of chemicals that are already made in all the cells of our body, the odds that some tweak in the amount of one of those is going to give us a transformative change of how long we live, I find it very, very difficult to believe that will happen.
That's a very interesting point about the substances that are already in the body, and that we shouldn't expect miracles to come from small or changes in the amounts or something like that. So that's an interesting point. I like that point. I would say, for as far as Sinclair, is that getting into this epigenetic reprogramming, I think I'll say, to me, sounds at least promising. That's at least an area that has seemed to me for some years has been a promising area. If he can actually make some progress, and he apparently has made some progress in animals with, what did he actually do? It was in something to do with vision. I don't know if it was macular degeneration or something. That actually was pretty good, and I'd like to see more of that. But I agree with you about the other stuff that when we're talking about.
In concept, epigenetic programming could be more encouraging than the small molecule intervention. The SIR2 framework just never clicked with me. The very first articles just didn't make sense to me ever.
Well, I'm sorry to say it did for me when I first read them. It made some sense, and I was caught up in the resveratrol thing, but it very quickly petered out, and it ended up that just in the way that Wolf says, and this was known back then, that the resveratrol really only works when you give it to rats or mice that are fed a McDonald's style diet consistently. Then it worked, then it did something, but it didn't even work with the regular diets that Wolf thinks are lead to too much obesity. So it basically, that whole stuff, as far as I know, the whole approach failed to produce real life extension.
There was huge hope in 2006. We were all at this conference waiting for the results. Is it going to benefit the regular ones, not just the ones on the high-fat diet? And in the end, the answer came out as a disappointment. I remember that was quite palpable.
Okay, so I want to just say one thing, and then I'll let Robin wrap up if he has a final comment, because we need to go over to Clubhouse. But I'd say of the things that are currently being worked on, and I didn't even mention everything. I mentioned a lot of the big things and asked about some of them, but there's a lot going on in the field. I would say that the one thing, if you're going to reduce it to one intervention that is likely, and at least this is my view, likely to bear fruit in the very near future, it's Senolytics. As a matter of fact, there is a Mayo Clinic trial going on right now with humans, so not just rats, because rats are, with some exceptions, not human. They have an actual trial with humans going on. It should be pretty close to ended, and we'll see with Ficitin, which is something you can get anybody can get off the shelf. Now, I'm not going to give you advice, because I have a PhD, not an MD, and my PhD is in psychology for that matter. So I'm not going to try to give anybody medical advice. You have to figure out for yourself. But this is something that somebody could try if they wanted to try something. This is an area where you actually could try it. And Ficodin is, again, I'm not going to make definitive pronouncements, not being a medical doctor, but Ficodin is pretty safe. It's been used by humans for a long time in small doses. Anyway, I'll just leave it at that. Senolytics, they're working, there are a lot of companies working on it, trying to come up with stuff that they can sell in the near term, and I think that's going to happen. And then that I hope will bridge us. I hope will give us part of the bridge that we need to, more extensive and comprehensive therapies, such as from stem cells. Okay, Robin, Dr. Mockett, what do you want to add here?
I have to play on what you said about rats, with some exceptions not being humans. I'm not aware of any exceptions, but I will say that humans, with some exceptions, are not rats.
All right.
There might be exceptions in the reciprocal case. I do want to echo, and for myself say the same, that you said, that I'm not medical professional, and if I were a medical professional, I wouldn't be the professional of the person I can't even see on the screen, because I just see your few pictures. So I wouldn't want to advise, but I do think the thing about guarding your weaknesses is general enough that I would put that out as advice. If I think about bridges, we didn't talk about it, and it's off topic, and I'd say it's a really remote possibility, but I like the idea of some further discussion one day about the whole cryonics field.
Oh yeah.
And so the idea that, well, now we can preserve some of our cells through spinal cord, through umbilical cord harvest at birth. I wouldn't mind if there were some cells of me in my original form, because one of the articles I read that really impacted on my thinking this year related to the amount of mutation that happens in cells, even that are not dividing, due to damage during gene use, gene expression. And so,
but Robin, how much of your cord blood is there still existing? In my case, I think it's none.
Right, it's none. So I'm saying
not going to help any of us here, not even Steve.
Right. What I'm saying is, it would be very nice, and that's where if I were bringing somebody into the world, I would probably look into trying to keep some as an act of generosity to the person I was bringing.
So any of you out there that are about to have babies might consider that. At least think about it.
Yeah, it basically puts you on. It puts some copies of your original self on ice for potential future use. Because what I took from the study of neurons was that all the cells that we're using are getting changed through mutations. So the average would look like about 15 mutations per neuron every year, which is making them different. We got to move over. We can keep talking, and we want to. And by the way, we probably should just bring you back, Dr. Mike, because we didn't really talk about your research, much less the cryonics. So we need to arrange to get around to your research. But let's anyway. It's time to go over, isn't it, Daniel? Time to go over to Clubhouse. Can I have 1 minute for everybody for the transition to the Clubhouse? The work I've been doing relates to rapamycin. There was a paper that came out just in the past 2 days from Linda Partridge's group. I've been working with flies from her group, been interacting with them for a while. I'm seeing a range of results related to rapamycin and diet. and other TOR inhibitors for longevity of life. So some of the same work, but I'm not funded on the same sort of scale. So I don't do the more complicated research, but the actual feed this chemical under this condition, does the animal live longer? I can do those experiments very well, and her group does them very well also. But we're seeing quite a range of outcomes. And so if I look at the idea that rapamycin is good, and I look at things where I've sometimes found that something someone said was good and I found it wasn't useful, I have substantial concern about rapamycin in safety under certain circumstances. And
all right, well, we'll have to get back. We'll have to get,
So-
we maybe be talking Clubhouse, but we may have to bring you
that's
back.
for the club. That's
that
for
one.
the clubhouse. And
And
anybody,
yep.
yeah, anyone
Because,
who wants to know what I'm doing
that's all.
in this, that's for off the just the whole world pre-publishing.
Oh, I see. You're good. I see. Okay. All right. Well, let's do that. Thanks everyone for coming. Steve, you know how to end us best.
Sure. Thanks for coming to the cutting edge today, and we will see you in 2 weeks. And thanks for Dr. Robin Mockett for being on the show.
Thanks, Rob.
Goodbye.